So we've got a zygote. The nuclei have met and fused. The centrosome and its microtubule array have brought them to the center of this very large cell. This zygote lasts a very short time, approximately as long as its first division. As soon as they finish replicating DNA, they'll enter first mitosis, beginning with prophase, one of the first of 5 characteristic stages of mitosis. During prophase, the nucleus is still intact, but the chromosomes have begun to condense. Meanwhile, the centrioles have fully matured and separate from each other as 2 new centrosomes. Each with their own array of microtubules. During prophase, the nucleation rate for microtubules at each centrosome goes way up, but the average lifetime of an individual microtubule goes way down. That is, the catastrophe frequency increases. So these microtubule organizing centers emit extensive, dense arrays, but of relatively short microtubules, many of which interdigitate between them, and these 2 microtubule organizing centers separate towards antipodes of the nucleus, opposite poles, that is, through the agency of a microtubule motor that engages with anti-parallel microtubules and tends to slide them apart. Meanwhile, the microtubule organizing centers remain stuck to the nuclear membrane surface through the agency of dynein, the same dynein that brought the pronuclei together. Next comes prometaphase, in which the nuclear membrane breaks down, allowing those microtubules to interact with the chromosomes. Again, at this point, the nucleation rate for microtubules is very high, but so is the catastrophe rate, meaning we get short, dense arrays, or dense arrays of short microtubules. Without the nuclear membrane, there's nothing to really keep the centrosomes from continuing to slide apart from each other, and the spindle poles are starting to separate spatially. Once these microtubules that have penetrated the nucleoplasm, or what was the nucleoplasm, have engaged with the chromatin, they begin a process of aligning those chromosomes at the midplane of the cell, which culminates in metaphase. So in metaphase, all of the chromatin gets lined up at the future plane of division. In fact, in animal cells, the plane of division is defined by the midplane of the mitotic apparatus, the microtubule-based chromosome sorting machine. Now let's draw in the centrosomes and the microtubules, which compose the mitotic apparatus, which is also called the mitotic spindle because of its overall shape. Microtubule nucleation remains very high, catastrophe rates remain high. So we've got a lot of short microtubules coming from each centrosome, but some of them are stabilized by their interactions with the chromosomes, and this thing here we call the spindle, loosely. And its structure is dominated by these bundles of microtubules that connect to each chromosome. the centrosomes are now called often spindle poles. That, of course, is a functional name at this point. They serve as the poles of this apparatus, between which the chromosomes are strung. Once the cell detects that all the chromosomes are aligned equally between the spindle poles, something happens. It's the next subject. Something happens to cleave apart the duplicated copies of each chromosome so that one goes that way and one goes that way at anaphase. During anaphase, these microtubule bundles that constitute the mass of the spindle, they shorten as chromosomes are drawn back towards the spindle poles on either side. There remain some number of microtubules that span the distance between the poles. These so-called interpolar microtubules have an important role in the development of the next stage in the process. At anaphase, microtubule nucleation slows down dramatically, but so does catastrophe frequency. Consequently, we have fewer microtubules initiated per unit time, but those that grow out from the centrosome tend to last longer, and so they reach further into the cytoplasm. Furthermore, as the chromosomes separate in anaphase, the spindle poles separate too in most cells. In fact, they're getting progressively further and farther apart as they go into telophase. Telophase is the part of mitosis when the cell actually divides itself in two and the nuclei reform. The centrosomes are now quite far apart from each other. In fact, they have adopted nearly the center position of the new daughter cells. The nuclear membrane reforms around segregated chromosomes in telophase. First as some little droplets, and then those droplets fuse together to make a single nucleus. What about the rest of the microtubules? Well, there remains some bundles of microtubules that one might think are a spindle remnant. In fact, these mostly consist not of the bundles, remnants of the bundles that connected to the chromosomes, but rather these interpolar microtubules here. The centrosomes continue to nucleate somewhat fewer microtubules, but those that they emit are relatively long-lived. In fact, many of them reach all the way to the cell surface. While I draw, why are they curved? Well, it is precisely because at this point, the cell is dividing. The cell surface is indenting in the cleavage furrow that will pinch down and partition two daughter cells, each having a copy of the chromosomes that started out in prophase by condensing. Those are the stages of mitosis. And of course, lest I forget, there's interphase. when nothing interesting is happening. The centrosomes do not organize mitotic apparatus assembly alone. In fact, the kinetochores dominate key aspects of spindle assembly. Kinetochores are the platform on each chromosome to which microtubules attach during assembly of the mitotic apparatus. Now, we're going to consider for just a single chromosome in isolation here, a sort of double bunny configuration that appears so often in diagrams like these. The kinetochore is a discrete macromolecular assembly, typically at one location on the chromosome, built around a region of the chromosome called the centromere. This plate here is the morphologically visible structure, the kinetochore, which serves as a platform for microtubule attachment. And these things are connected in a replicated pair of sister chromatids. This is a chromosome. Each of these is a chromatid. Again, the kinetochores serve as platforms for the capture of microtubules during spindle assembly and the stabilization of a bundle of microtubules to which they're attached to their respective spindle poles. This bundle is referred to as a kinetochore bundle or a kinetochore fiber or simply a K fiber. Now let's take a look at what's inside the kinetochores. Kinetochores do a number of things besides simply capture microtubules. First of all, they measure the tension that the chromosome is under. When a chromosome is properly attached to the spindle, it's under tension from both of these microtubule bundles, and the chromosome contains a sensor for that, and it also contains a signaling hub that tells everybody else what it's feeling. And that's the sense in which kinetochores in part govern mitotic apparatus assembly and function. The kinetochore, in a way, is the selfish gene's way of not getting left behind, okay? So let's take a cartoon look at not the structure so much as the functional parts of a centrosome. First of all, there's that plate paired with the plate on the sister chromatid and connected by something that allows them to sense tension between them. Let's just say it's a spring. Built onto that spring is some sort of signaling hub, something that will send out a signal that can be heard from far away. At least we hope it can be. Again, one of the principal functions of a kinetochore is as an attachment platform for microtubules, and it happens that kinetochores have a sort of a sliding clamp. In fact, in yeasts, that sliding clamp really looks not unlike that and accommodates a single microtubule. Instead of a great big bundle, they've just got one microtubule per kinetochore. They've also got motors. I'll draw dynein in here again. Unattached kinetochores happen to have plenty of dynein on them, which of course, does what dynein does. It captures a microtubule and walks towards the minus end. Remember, the minus end is going to be out there at the spindle pole, where ultimately this chromosome ought to end up. That's supposed to be a pair of scissors. Each kinetochore also has to have some way of pruning aberrant connections. So that's what's inside a kinetochore: a tension sensor, a signal generator, something with which they can attach to microtubules and hang on to them as they grow or shrink, something with which they can clip off microtubules, destabilize ones that aren't engaged constructively, and then something that will capture them and pull on them. Something about engagement of microtubules with the kinetochore stabilizes the microtubules of the K fiber so that they are much less likely to undergo catastrophe. They don't grow as fast as microtubules growing into the cytoplasm would, but they still grow. And that leads to the following interesting outcome. Let's start with the metaphase spindle. I'm just going to draw in the microtubules and then describe an experiment. Here are the K fiber bundles making up one half of the spindle. And of course, what we call astromicrotubules around the spindle pole, continuing to reach out. On the other side, I'm going to do a little experiment. I'm going to mark a band along the kinetochore microtubules on the other side. This experiment has been done several ways, but one of the most revealing is to use a dye that can be converted from a non-fluorescent form to a fluorescent form with a pulse of ultraviolet light. And thus, I'll draw the now marked K fiber sections in red. So we can watch over time what happens to these. And it's an interesting exercise to think, well, what are the possible outcomes? So if we just diagram now the brightness of our mark with distance from the poles, then at this point where the mark is made, the brightness is a very well-defined peak, right? Now what's going to happen to it over time? Well, it depends on what the microtubules and the K fibers are up to. I'll tell you what actually happens. Over time, the mark remains more or less coherent and it moves toward a pole. Okay, it spreads out a little bit. But the thing is, imagine what else could have happened and what the interpretation would have been. It could have been the case that over time, that mark simply faded away and diluted. That would mean that the marked tubulins composing the kinetochore fibers were simply spreading apart diffusively. Or it could have been the case that the mark simply faded away in place, or something like that. These would all have different interpretations, but the true one is what I drew initially. All of these microtubules are coordinately moving, flowing from kinetochore to spindle pole. This poleward flux within the K fibers is one of the factors which exerts tension upon the kinetochores when they're lined up correctly on the spindle. Now, after decades of study, still no one can tell you exactly how the chromosomes end up right in the middle, what forces act upon them. But there are a few components that are pretty clear, one of which is the one I just mentioned. The flux of kinetochore microtubules toward the poles. In the metaphase spindle, that's pulling each still connected pair of sister chromatids that way and that way at the same time. Right? Another thing that's clear is that unattached kinetochores have dynein on them, which connects to those microtubules and pulls poleward as well, because remember, all the minus ends are out there at the poles. So dynein also pulls poleward. Now, an interesting observation, if you look at chromosomes as they're lining up relative to, say, a spindle pole, before they've made all their attachments, it often looks as though the chromosome attached by its kinetochore, let's say it's still unattached by the other spindle pole, it looks like it's getting sort of blown backwards in some sort of a wind. And it turns out, yes, there's dyneins at the kinetochore pulling toward the poles, but along the chromosome arms, there's another microtubule motor that tends to walk towards plus ends. Consequently, chromosomes experience what's described as a polar ejection force localized to their arms. Something is blowing them away from each spindle pole. This is often referred to as the polar wind. Finally, tension across the kinetochore pair regulates microtubule attachment. These add up to an approximate mechanism in which the chromosomes are blown, pushed, pulled back and forth until they're equidistant from the spindle poles. Again, this is the point where all of these forces would equilibrate, and this condition is detected at each kinetochore by something that measures tension, and each chromosome in effect yells out through the signal generator, wait for me, wait for me, wait for me, until it detects an appropriate amount of tension across its kinetochore pair. Once all the chromosomes shut up, the idea goes, that's when the cell triggers anaphase by snipping apart the sister chromatids at the kinetochore. I want to narrate one elegant experiment that addresses this second point that I've added here, that tension across the kinetochores gates the progression of the cell cycle from metaphase to anaphase. So let's consider a metaphase spindle again. And I'm going to highlight just one of these chromosomes in here in blue because Once I get the kinetochore fibers drawn in, because someone's going to come along with a needle and hook that one and yank it out of the spindle, breaking its kinetochore fiber connection to the spindle poles. So the next state, the next picture in time looks like this. We've got a detached kinetochore floating out there away from all its compatriots, detached from the mitotic apparatus. Bad news. The idea of this tension sensor in the kinetochore that sends out a signal is that this chromosome now yells out loudly, wait for me, don't proceed to anaphase without me, get me back into the spindle, please. Okay, so roughly the idea is that if the cell waits to proceed into anaphase, that gives the mitotic apparatus a chance to recapture that chromosome. Say, if one of these astromicrotubules manages to grow out that far, then of course this chromosome gets captured, experiences a pull back toward the mitotic apparatus. Let's draw a kind of an intermediate stage here where the polar wind is also in effect. It'll get drawn close to one spindle pole, but not all the way there, because of course the pole dragging it toward the spindle pole is eventually going to be balanced by the push driving it away. So we'll get to a point where here's all the other chromosomes waiting patiently, still attached to their kinetochore fibers. And here's the stray. And I'll draw its connections here in a special color, because the immediate outcome, the desirable outcome, is that another microtubule, perhaps extending far across the metaphase plate, might reach out, grab it, and then this chromosome will eventually undergo the sort of push me pull you that chromosomes do to find their way to the midplane again while the other ones wait. And once it gets there, it will stop emitting this alarm call, which it's still emitting here until it makes an attachment to both sides. Once it gets there, it'll shut up, stop saying wait for me, and the cell will proceed to anaphase, and all is well. At least in many small cells, one stray chromosome really can hold up the cell cycle from progressing into anaphase until recapture gets this checkpoint satisfied. Now, what happens if at this point the mean guy with the needle comes back and pulls on that chromosome before it finds its way back to the spindle midplane? So here's all the other chromosomes. Now, the experimenter in this case is very careful not to break it away from its attachment, but simply to exert tension on it. Well, what happens is that tension will satisfy the checkpoint. This chromosome will stop releasing a signal that says, wait for me, and the cell will proceed to anaphase. And what happens to our stray? Anaphase onset means that the two sister chromatids are separated from each other. And that connection will get one of those, perhaps, to one spindle pole, but the other one will be lost or incorporated into a cell randomly. And that could be quite a disaster. This experiment demonstrates, first of all, that, again, one stray chromosome can hold up the entire cell cycle, and second, that tension alone can satisfy this checkpoint. The cell has no way of distinguishing the experimenter's applied tension from the tension that would be applied by engagement with two spindle poles equally.